So, welcome back to our last presentation, and I see Thomas is already ready to go. Let me briefly introduce our last speaker today. We're very excited to have you and are really curious to hear from you because it's a topic that we haven't discussed yet, but that we have as groups been discussing for many years in various shapes and forms and constellations. Let me first introduce who you are, because we haven't met personally yet. I don't think anybody in the room has yet met you, so it's wonderful to make your acquaintance via this platform. You are a professor of anesthesiology at the medicine and physiology at the David Jeffens School of Medicine at the University of California, Los Angeles. You work on epigenomic mechanisms for cardiovascular diseases. I can reveal to you that we have a professor of cardiovascular sciences among us, and I think he's still there, so I'm sure he's listening carefully. And with the goal to discover new therapies and cures. So that means you are also desperately trying to find new breakthroughs for diseases we haven't been able to cure yet. And I very much look forward to hear this from a medical person. And I think with that, the paper that struck me when I did a little Google search for you was Genomes, Proteomes, and the Central Dogma. So you take us directly back to the Central Dogma and some of the discussions we had yesterday. So curious to hear what your take on this is, and I'm sure we will have lots to discuss afterwards. So with that, I will give you the virtual platform to you. You can unmute yourself, or is he unmuted already?
Yeah, I think I'm unmuted. Can you hear me?
Yes, everything seems working well.
Okay, wonderful. Well, thank you for the introduction, and I really want to thank all of you for inviting me to this conference, in particular the organizers, and specifically Dr. Siguna Muller. We met also virtually years ago when we exchanged some emails about some of our respective work, and it's been a great pleasure for me to participate in this conference. And I have to tell you, I have no idea. I had no idea what to expect, and I had no idea whether what I put together is going to be interesting to anyone. So hopefully this is something that would strike some of your interest. And so I'm in Los Angeles. I'm at UCLA. We're a basic and translational research lab. And so what I thought I would do today is kind of moving from some of the other talks that I listened to in the middle of the night here on my time, to tell you a couple stories, very brief ones, about some of the work we're doing to understand how genomes are involved in human disease. And so I wanted to start with this slide. Actually, I put this in just at the end of Dr. Soto's lecture here, because I thought a lot of the discussion there had me thinking again about these cognitive biases with which we all come to studying biology. And one of the things that we often talk about in the lab and with our students is sort of the difference between evolved systems and engineered systems, and how both of these can be extraordinarily complex. But oftentimes it's the cognitive biases we have from thinking about the way human beings build things, and then we bring those over to the study of the natural world. So if we look at Linus Pauling, or my dream car here, a 1967 Ford Mustang, both of these are very complex. They both have emergent properties. That is, they have features that can't be reduced to any individual feature within either of those complex systems. They're both controlled by networks. I would argue one of the major ways that we start to understand the differences between them is talking about redundancies. So you can think of the human eye or the space shuttle. They're both very complex. A contrast is that evolved systems tend to be versatile. redundant, and therefore inefficient. So by definition, evolved systems respond to change. This is something several of the other speakers have been talking about. And redundancy exists at all scales of biology. And one of the things I'm going to talk about today, the principal thing I'm going to talk about, are the scales of complexity within the structure of mammalian eukaryotic genomes. And we're going to talk about that redundancy and scaling phenomenon. In contrast, engineered systems are rigid. They lack this redundancy, but that makes them very efficient. Okay, so the 67 Mustang that I'm lusting after cannot easily adapt to operation underwater, right? So it's very rigid, although it is very efficient. And biological systems exhibit self-similarity at multiple scales, okay? Nature selects for something that works, and here's this terrible word that we touched on in the last talk. This mechanism is used repeatedly in different systems. And I really like that and agree with the discussion about the problem with this word mechanism as a cognitive bias. And it's something that we try very hard to strip out of use in the way we think about our work. So the question that my lab is interested in asking and hopefully answering is, how does one genome create hundreds of cells? So the genome, the sequence of the DNA is the same in all the cells in the bodies of all eukaryotes, but the packaging of that genome, the structure, and therefore the function of the genome is dramatically different between different cells, okay? So the cell type we're interested in, it was also touched on in Professor Soto's lecture, is the heart, the cardiomyocyte. So this is a microscopic image of a cardiomyocyte, and the green here is decorating the myofilaments, and the blue is binding to the DNA. So this is a stain that basically allows us to identify where the nuclei are. And so the focus of our lab is really to understand what's going on inside the nucleus and how is the genome packaged differently between the different cells in the body. The other interesting thing you can see about this cell is that it has two nuclei, which is a common feature of mammalian cardiomyocytes. We can talk about that a little bit more later. Okay, so let's set the stage here for what we're going to be talking about in terms of a structural hierarchy of how the genome is packaged. So we can think of this at different scales. Let's start over here at the nuclear scale, bottom right. So if you look at the entire nucleus, it's been known for decades, almost a century, that the chromosomes in the interphase nucleus, so when the cell is not dividing, they form these territories. So they kind of segregate themselves within the nucleus. The specifics of this are very poorly understood, but this has been known for quite some time. And then if we jump way over here to the other side, to what I've called the nucleosomal scale, this is just probably the simplest way to think about this is just individual genes on the chromosome. So the way that these are sort of regulated, okay, whether a gene is turned on or off, has also been studied extensively now for well over 50 years. And a lot of the information about how genes are turned on or off at this scale is fairly well understood. But what's not understood, or what has only recently become apparent, is this, you could call it supranucleosomal scale, so above the scale of the individual packaging unit of chromatin, but below the scale of the whole nucleus. How has evolution packaged this enormous amount of DNA into the nucleus, okay? And so we have to, I think, in thinking about this, address, I think, what is the elephant in the room about cognitive biases in biology. And I know this is something that's been talked about in this meeting as well. And that's the central dogma of molecular biology. And I'm drawing it out here as my college professors drew it out for me on the board when they told me that no study of biology can begin without a reiteration of the central dogma. And the problem is that as I've written it out here, and as I oftentimes in haste, describe it to my own students, this creates a cognitive bias in terms of the way that we think about the function of biological organisms, as many other speakers have iterated. And that's the idea that it's this unidirectional flow from DNA to RNA to protein in terms of biological function, okay? But if we look back at the way that Sir Francis Crick originally articulated the central dogma, it was much more nuanced than the way that I draw it out for my students, okay? And that's that the DNA information flows through RNA and onto protein, and then it doesn't flow in the opposite direction, okay? And what he meant here was the transfer of information in terms of between biomolecules, but also in terms of heredity, okay? This was one of the original times, I think, from one of his notebooks when he wrote it down. And then this is from a paper also from Dr. Crick in, I think it's 1970. I can't see the top of my slide here. Yeah, 1970, where again, he talks about this is a model for the flow of biological information, all right? And he talks about these other types of things that would be precluded by this model, right? And I think we like to remind ourselves of the appropriate use and the appropriate understanding of the central dogma as opposed to the inappropriate way that I drew it out on that first slide. And as such, there's no problem with the central dogma of molecular biology. There's only a problem with how we teach it and how it serves as a cognitive bias. And so what Dr. Crick's original articulation referred to was the accurate transmission of information between biomolecules, including for hereditary purposes, okay? What it does not concern is whether and how biomolecules, the many different types of biomolecules, RNA, DNA, protein, carbohydrates, lipids, and others, can modify the behavior of each other. It does not mean that DNA controls everything. And this reality is central to the way that we're pursuing the question of that I asked at the beginning of the lecture, which is how one genome, one sequence of DNA can encode hundreds of different cell types in mammalians. Okay, so we're going to be focusing on understanding this structural hierarchy, and I'm going to tell you more what I mean by this here in a second. So another very influential model in terms of thinking about this was articulated by Conrad Waddington, and this was originally published in 1957. I think many or all of you are familiar with this model of the epigenetic landscape. And what Waddington was talking about here was a developmental landscape, which he modeled as this ball that runs down through these pathways and becomes more committed. So this is an egg that becomes more committed to a somatic cell. And you know, this model was really genius, not only in terms of being a very sticky idea. So once you see it and understand it, it's kind of so intuitive that you never forget it. But really what Waddington predicted at this time, decades ago, was many of the things that we now understand to be the case. So during differentiation, there are these bifurcations, there are these decision points where a cell becomes more committed. And the energy landscape serves to restrict the cell or attempt to retain the cell within that energy landscape. But it's not permanent. Okay, so we now know that cells rarely under natural conditions will transdifferentiate. So if you imagine balls down here, that cell or ball can transdifferentiate to another state. We can use genetic engineering to force the ball back up this valley against its energy gradient to take another path or to go back up and become, in fact, pluripotent. Okay, so we now know that we can induce pluripotency from somatic cells. The other thing that Waddington predicted from this, and this is the other side of this diagram, which is often not known, is really what we're trying to study when we're talking about the structure of the genome, which is what's going on under that landscape. Okay, and Waddington called these guy ropes, and he connected them to the genes. And basically what he was already essentially predicting here is that this epigenetic landscape, as he called it, was maintained not by the direct action of individual genes, but by the constellation of interactions between these genes and other factors that serve to create and maintain this epigenetic landscape. Okay, so let's talk about, I lost my cursor, bear with me, the structure of the genome. Okay, so back for a second. If we look at individual DNA, it's wrapped around these protein complexes called nucleosomes. So right there in eukaryotes, you can't have function of the genetic material without proteins to package that genetic material into these chromosomes then in the nucleus. And if you open any molecular biology textbook, you know that the mitotic chromosomes form these beautiful things you see here at the bottom. But this is only when the cell is dividing. What's going on when the cell is not dividing? So each mammalian cell has about 2 meters of DNA into it, okay, in it. And the nucleus, depending on the cell type, is a few microns across. So this is an enormous compaction event that nature has figured out, and figured out how to do it reproducibly and very precisely in different cell types. So how would human beings do this? How would an evolved, how would an engineer solve this problem? Well, I really like baseball. Okay, so I like the analogy of the baseball. So it turns out that the way a baseball is made is there are about 300 meters of string that are wrapped around this cork and rubber core, all right? And so this is a pretty incredible compaction event in and of itself, all right? It's super compacted and it's very ordered, right? But we know from looking at microscopic images and also from sort of common sense, this is not the way that evolution has solved this problem. There are discrete neighborhoods that exist in the nucleus that are very different than just wrapping string around a ball, okay? Here's an example of that. So these are 3 different nuclei from 3 different cell types in the same organism, happens to be a mouse. So a fibroblast, a neonatal cardiomyocyte, and an adult cardiomyocyte. And you can immediately appreciate that the same genome in this organism, it also turns out the fibroblast is from the heart, in addition to the myocytes. So at different developmental stages of the same cell type, you have very different organization. In different cell types in the same organ, you have very different organization of the genome. Okay, so another model would be, okay, if it's not a baseball, what's another model that I could think of? Maybe it's like spaghetti. Maybe it's just all the DNA is sort of compacted in there in a very haphazard manner. This also doesn't seem appealing because we know that cells respond very specifically to stimuli from the extracellular and intracellular environment. So about over a decade ago now, a group in Boston led by Job Decker showed that the genome can be represented as a fractal globule. Okay, so they did this using cell lines. And what they showed, and I'm going to go into more detail about these experiments in a second when I show you some of our data, is that the chromosomes basically segment themselves in 3 dimensions into these neighborhoods, okay? And they have, like all fractals, repeating structural elements that exist at different scales, okay? They also noted that there are no knots in this, okay? So unlike a bowl of spaghetti, the spaghetti can get wrapped up. Evolution has figured out a way to structure the genome in the cell such that the DNA doesn't get entangled, okay? So speaking teleologically, that would be a good thing if you need to reproducibly fold and unfold this extraordinarily complex structure. So probably a better food-based analogy, and I know it's the afternoon there in Bern. I wish I was there with all of you. You're probably getting hungry. Probably a better food-based analogy is broccoli, okay? In that broccoli has some of these fractal properties that are shared by your genome. But what about this key question of cell type specificity and the changes that happen with disease? And this is really the focus of our research. So we're actually interested in the function of the heart and understanding how all of the factors that determine the normal and pathophysiological behavior of the heart are integrated in the cardiomyocytes and other cells in the heart, and also in the many other cell types in the body that regulate the health of the heart, including the blood vessels, including the neuroinflammatory system, the autonomic nervous system, which directly innervates the heart, the effects of hemodynamics and cardiac mechanics, in addition to the electrophysiological properties of the heart itself, the inputs from diet and metabolism, and also the role of genetics. And so our approach is to think of the epigenome now. So the genome, I'm going to define the epigenome as the DNA and everything that binds to it, and all of those processes that in binding to and regulating the genome control its structure and thereby differentially enable all of the varied functions of all the cells in the body. And so we think of the epigenome as this great integrator of all of these different inputs from different cells in the body, and also, of course, with genetics. Okay, the work I'm going to tell you about is done by in large part by these two individuals, Dr. Doug Chapsky and Dr. Manuel Rosa Garrido, who are I work with here at UCLA, and also we collaborate with Frank Albert and members of his team. So when we think about the structure of the genome, we and others have discovered that they form these topologically associating domains, and I advanced too fast there, sorry. But basically what this means is there are some local regions of genes and non-coding regions that interact more with themselves than they do with neighboring regions, okay? And so this is where the broccoli analogy comes in, okay? And this is very important for several reasons, because it suggests that this area that has privileged interaction could be a neighborhood of shared regulation. So you have a structural similarity that leads to a functional similarity. And same thing goes with this nearby neighborhood right here, but also that these two are segregated from each other in 3 dimensions, okay? So they can be differentially regulated. So if we look at this in the cardiac nucleus, first we can look at individual chromosomes. So here I'm just showing you chromosome 1 and chromosome 2, how they localize inside of a cardiomyocyte nucleus, okay? And what you can immediately appreciate is we can think of these as sort of domains of a protein structure. We can think of them as limbs of the body, right? So there are certain areas that have specific and reproducible interaction. There are anatomical features, okay? And there are regions that do not interact with each other, okay? And we think that this information is as important as what does interact with each other. So these are 2 chromosomes. What if we look at all 19 chromosomes, in this case in the mouse, okay? We leave out the sex chromosomes here, and this is a busy slide, but I want to use it to illustrate a couple points of what we can do with these models. Okay, so each ball you're seeing here is one of those topologically associating domains. It's one of those heads of broccoli in your genome, okay? Because this is based on DNA sequencing information, we know what each one of these dots are. So we know what the genes are in each of these areas, okay? Because we make these measurements in cells, this is not just DNA sequence. It's not just determined by DNA sequence. It's determined by how the DNA interacts with the totality of proteins, lipids, and RNAs and other molecules that are inside of the nucleus. Okay, so this structure is a recapitulation of that endogenous behavior. Okay. What I'm showing you here is how one of the ways that we can use these models, which is to map onto them other measurements that we can make of the cells. In this case, we're measuring changes in gene expression using a technique called RNA sequencing, okay? And the difference between the one on the left here and the one on the right is this is the same genomic structure at two different stages of disease. One is an early stage disease, okay, of the heart. And then the other one is that's the left side. The right side is late stage disease, heart failure. And so what you can appreciate is that at different stages, of disease in the same epigenome, we have different features that materialize as a result of the interaction of the other aspects of the cell and the extracellular environment, which induce changes. In this case, we're mapping gene expression, okay? And so, for instance, here on the left side, you can see there are these large contiguous green areas representing areas of downregulation. And these same areas of this structure, if you look over here on the right, many of these now, no DE means no differential expression. So at this later stage of disease, they're no longer differentially expressed. So you have a structural feature that now has a functional change as a result of the advancing of this disease. Okay. One of the questions we wanted to try to answer with these models is to understand whether these structural features can tell us something about the way evolution has solved some of the problems that exist in single cells organisms like bacteria that also exist in mammalian cells, an example of a eukaryote. And that is, how are the genes that need to be turned on in a given situation coordinated? Okay, so I just showed you some of those structural features that we think are one underpinning of that. But the way that evolution solved this problem is in bacteria is it lined up all of the genes on the same chromosome right next to each other. So if you have a response to sucrose in the media, then all of those genes are right next to each other and they all get turned on. But it doesn't work like that in eukaryotes. So what we thought is maybe the three-dimensional organization of the genes is a partial solution, okay? So the prediction that we had when we did this was that we would have two types of behavior, okay? We would have one where the majority behavior was an increase in expression for a given neighborhood. So a given head of broccoli would behave in one direction only. And then that would be the ones that were increasing expression. And then we'd have another group that would have a majority in the opposite direction, would be decreasing. But then we thought about this some more and we said, well, there's also the possibility where you had the one genomic neighborhood where you had the exact same number of genes that were upregulated as were downregulated. And maybe this would be something that evolution had selected for, to have a more plastic region of the genome. We thought that this was unlikely, but we recognized that these were three independent predictions of this analysis. So what did the data show us? Okay, that's what I'm showing you here. It's a complex slide, but it has a very simple straight take-home point. So each of the different colored regions here, if we move from left to right, are the different chromosomes, 1 to 19. Again, this is in the mouse. And this is at an early compensatory stage of disease, okay? And each one of the dots represent one of those structural neighborhoods. So you can see that there are quite a few that are, these are neighborhoods with majority increased expression, okay? However, we had much more with majority decreased expression. And I'm gonna come back to this at the end of the talk, because we think that this sort of integrated structure function behavior has allowed us to learn a novel property of the way genomes function. But very interestingly, we in fact saw quite a few of these structural neighborhoods with perfectly balanced change. That means that this region of 3 dimensions has the exact same number of genes that are upregulated that are downregulated. And the way we interpret this is that these are these plastic areas of the genome that are primed to move in either direction. Okay, this is basically just showing you the same analysis at that later stage disease. So once the animals have progressed longer into disease, you can appreciate that the ones that are in this, what I called plastic sort of state, there's a decreased percentage of them there, but there's also a decreased percentage of the change overall, which if you remember from the previous model I showed you, was also what we saw in terms of total gene expression. Okay. So is there another way that we can examine these discrete functional compartments in the cardiac nucleus? And are these things specific to cardiomyocyte nuclei, or do they exist in other cells? So this is an example of some work done by the Kremers, which have really been pioneers in the area of endogenous genome structure. And what you can see here, what they've done in this experiment is use different dyes. So you're looking at a nucleus here. This is a single nucleus. And each of the individual chromosomes is labeled with a different color dye, okay, or different pair of dyes to elicit a different coloration of these chromosomes. And you can see that these chromosomal territories can be recapitulated by microscopy. So the things that I just showed you that are based on sequencing, you can also see them by microscopy. So here's some other data from our own work, which basically shows that there are different neighborhoods within the nucleus. So again, each of these are individual nuclei that are labeled with different markers. This is lamin, which marks the nuclear periphery. And you can see there's this specific modification to the region of the genome that causes that region of the genome to localize to the nuclear periphery. Okay, and this is just showing all the DNA, and this is a merge of all of these images together. So we wanted to ask the question, if we look in disease, are there structural changes in the localization of genes within the nucleus? And again, bring this back to this thinking of, okay, can we just explain everything by a transcription factor goes into the nucleus and there's a change in gene expression, right? Or is it that the DNA has to be packaged with the totality of other proteins, RNAs, and lipids in the nucleus to create this specific substrate that allows for cardiomyocyte or neuron or hepatocyte? If that latter model was true, then we should be able to detect structural reorganization of specific genes in the nucleus, of specific regions of chromatin, okay? And that's what this slide shows here, okay? So if we just look here, here the blue is all the DNA, all right? And the red dots are specific genes. And what we're quantifying here is the change in localization of these genes between the healthy condition here on the left and the disease condition here on the right. And what we found is that there's a relocalization of these genes in three-dimensional space from away from the center and towards the periphery when those genes are silenced, okay? Suggesting that there's a specific structural neighborhood responsible for this silencing. It's not just a matter of is the gene on or off. There have to be some other protein factors there that are responsible for this structural reorganization. Likewise, this gene turns on and it moves away from the nuclear periphery, okay? These two genes don't change in expression in the case of this gene, and this gene is never expressed in cardiomyocytes. And so they have a very different structural orientation and do not change their association with the periphery. Here's another example. So we can identify these structural features inside of the nucleus. So this is an example here. Again, all of the DNA is stained in blue. You can see that this is a very specific organization within the nucleus. And this specific gene shown here in red, I don't know if you can see it, is localized to this internal feature. So rather than being localized to the periphery, it's localized to what we call this central region of silencing, or central heterochromatin center. Okay, and so we found a logic for how different genes are structurally reorganized concomitant with their being turned on or turned off. So some genes, they change their association with the nuclear periphery. Some change their association with these central heterochromatin centers, and some do both. Okay, and this is just to show you. So again, I was showing you healthy and diseased hearts there. Here we're looking in the same model of heart disease, but now we're looking in the liver. And what you can see is there's no reorganization of these genes in the liver, which shows that this is cell type specific. So in the last few slides, last 2 to 3 minutes, I want to show you, I want to extend this theme about how chromatin structure differs between cell types. So how does this complex molecular origami inside of each nucleus, how has evolution used this to create these different cell types? Okay, so I think one of the most striking observations of this, certainly the most striking one that I'm aware of, was originally reported by Solove et al. And this is also work with McCremers, whose work I showed a second ago. And what they did in this study is they looked at the organization of the genome inside of the rod cells. So these are the light sensing cells in the eyes of nocturnal versus diurnal animals. So of course, nocturnal animals have evolved to see at night, to see in very low light conditions, whereas diurnal animals have evolved to do the opposite. What they found, to make a long story short, in this paper, by looking at a large number of different species, is that you can see here, this is a schematic representation of the genome structure in the mouse and the rat, which are nocturnal, versus the monkey, horse, pig, chipmunk, and tree shrew, which are diurnal. Evolution has selected for a chromatin organization pattern in the rod of the eye, in which the nucleus itself actually acts like a lens. So the DNA is organized in a way to act like a lens, to focus light. Okay, and this only happens in nocturnal animals, not in diurnal animals. So this is something that's really counterintuitive. If we think of every function of the genome has to be by the transcribed genes going on to make proteins and doing whatever they do, this sort of evolutionary adaptation sort of really challenges us to think about that in a more holistic way, because it is definitely true that this organization is as a result of the function of proteins that are specific to these cell types. But evolution is selected for this really bizarre and quite amazing phenotype. And this is just another example of that, again shown schematically here, that this develops over time. So this is a temporal development in the mouse here of this structure. So it's not something that is the same throughout different developmental stages. So in our own work, we've tried to ask similar questions about whether myocyte chromatin is somehow specified for the unique functions of a myocyte. To do this, we've compared some of our structural models with those that we generated using data from other labs, in particular on hepatocytes. And you can appreciate if you look at, so this is the same genome, excuse me, from the same species, it's a mouse. The ones on the left here are individual chromosomes are colored differently. The ones on the right are colored for active versus inactive regions of the genome. And you can appreciate that evolution has structured the same genome very differently in these different cell types in the same animal. okay? And if we look at this even further, if we look at cell type specific genes, so genes that are expressed, genes and proteins, in this case, that are expressed in myocytes in the heart that are not expressed in the liver, or vice versa, ones that are expressed in the liver and not expressed in the heart, we can see that there's a specific organization of structure around the ones that are cell type specific. So the ones that are specific for the heart have a unique structure in the heart, and they lack that structure in the liver, and the same situation for the liver genes. And so we interpret this as genome structure being more precisely regulated around genes that are involved in specific cellular functions. And of course, this structural regulation comes from all of the proteins and non-DNA components that are bound to the epigenome. We also found that if we look at the orientation of individual chromosomes in terms of being active or inactive in the heart and liver, you can see that they have this very different pattern. So the heart has this pattern whereby the active regions are preferentially localized toward the interior of the cell, as I showed you with the microscopy data. And the inactive regions down here, what we're calling compartment B, are preferentially localized toward the outside of the nucleus. All right, whereas in the liver, it looks pretty flat. There's no differential compartmentalization in terms of looking between the center toward the periphery of the nucleus. Furthermore, if we look at individual chromosomes, so this is again looking at the exact same chromosome in two different cell types, the red is the heart and the blue is the liver. You can see there are sometimes where the same chromosome localizes in almost the identical place within the nucleus. But then there are some times where it's poorly correlated, like here, or even anti-correlated here. Again, suggesting that the solution that's been arrived at for packaging the genome in these different cell types is very different. Of course, all the models I've shown you so far are, these sort of globular, spherical models. But as I showed in the very beginning, myocyte nuclei are not spherical. So one of the things that we're doing now is trying to improve these models by combining DNA sequencing data with microscopy measurements to get structural constraints for the building of these models. Okay, so this is our current model of a myocyte nucleus. So it has a lot of advantage over the other one that I showed you, in that, as I just said, it's structurally constrained based on the known anatomical dimensions of myocyte nucleus. It has a lot of limitations because even though we build this model on the basis of measurements that are made in the presence of all of the proteins and RNAs that are in the nucleus and responsible for chromatin structure, those features are not rendered in this model because we can't capture them with the sequencing techniques we use. Okay, but this is what we work from to sort of now start to understand the different behaviors of individual structural neighborhoods, individual genes or groups of genes, the way they behave inside the nucleus, inside the functional organism. Okay, so this is an example here of two different renderings of the model I just showed you on the previous slide, and these are two different copies of the same gene. And we can now understand the endogenous structural neighborhood in which these genes are functioning, in which their transcription is regulated. And we can ask questions about how those neighborhoods change in the context of disease. And this is really, we think, going to be critically important for understanding new treatment modalities, but also for coming up with new ways to characterize the progression of disease and to predict how it's going to develop. If we can show that chromatin is this integrator of all of these different environmental and genetic stresses that we know contribute to disease progression, if we can better understand that integrator, then we can use it to predict and treat disease. And so the take-home points then are that eukaryotes package their genomes in an ordered manner that enables precise and reproducible gene expression. Genome structure function relationships underpin normal development and also the response to disease. And as I talk about at the end, genome structure can perform these, what you might call meta functions, to control gene expression and phenotype. I think the most striking example of that is the example of the chromatin in the nocturnal versus diurnal rod cells. But I think there are other very interesting examples of that in terms of how evolution has shaped the chromatin in a way to specifically enable the behavior of that cell type. And lastly, we think that understanding how the genome is packaged within the cell may be the key to engineering novel strategies for combating disease. And that's all I wanted to say. This is a picture of the group recently, and I'd be happy to take any questions. Thank you very much.